Hello, welcome back to my channel. My name is Ashmita and I'm currently doing a series on the coronavirus pandemic known as Living in the Pandemic. This is the third episode of the series, and in this episode I'll talk mostly about treatment for COVID-19. If you guys haven't seen the previous two episodes, I would highly recommend you guys to check that one out first. So I'll leave the links down below in the description. Currently, there are no vaccines or medicines available for COVID-19, although there are vaccines that are currently in the process of being developed. However, the vaccines are not expected to come out until at least by the end of this year. Therefore, the current treatment mostly consists of antivirals. So in this episode, I'll be talking about the current drug therapy, how vaccines work, vaccine development in general, vaccine development for COVID-19, and finally, drug repurposing. Currently, there are 4 different types of drug therapy that are present. The first one is RNA therapy. RNA therapy basically modifies or provides RNA to patient cells. And there are 2 types of RNA therapy. The first one is antisense therapy, where they basically break down dysfunctional or harmful proteins in the patient's body. And mRNA therapy basically builds up functional proteins. The second type of therapy is known as cytokines therapy. So cytokines are low molecular weight protein functioning as chemical signals for the immune response. So what this therapy does is that it manipulates the immune response to build appropriate immune cells. The third therapy is known as small molecules therapy, and these are basically molecules that show a significant inhibition towards key proteins in similar coronaviruses. So candidates taking these drugs show inhibition towards viral enzymes, including protease. Finally, there's also antibodies therapy, and antibodies are basically produced by the body's immune system, and they bind to specific markers. So this therapy uses monoclonal antibodies, which are antibodies that are made in lab, and they are used to again build appropriate cells for the immune system to respond to the disease effectively. So the next topic that I'll be discussing is how vaccines work. Vaccines work by training the immune system to identify and combat pathogens effectively, and antigens are basically molecules that trigger the immune response. In general, there are 2 different types of vaccines present for both viruses and bacteria. The first one is known as live attenuated vaccine, where basically the vaccine contains a weaker version of the virus or bacteria. And because it is the closest to the natural infection, it is an effective teacher for the body's immune system. An example is the chickenpox vaccine. The second type of vaccines that are available in general is known as inactivated vaccines, and these vaccines contain the inactivated form of the virus or bacteria. And because it contains the inactivated form, it usually needs multiple doses for someone to become fully immune to the disease. For the SARS-CoV-2 vaccines, there are several types of vaccines, which include viral vector vaccines, protein-based vaccines, as well as nucleic acid vaccines, which include DNA and RNA. The next topic that I'll be discussing is vaccine development in general. So vaccine development is a very lengthy process because it has several stages, and therefore vaccines take a really long time to be produced. The first stage is to usually identify a synthetic or natural agent for prevention, and then researchers usually do cell culture or tissue culture in order to see if it really works to provide immunity. The third step is when private companies submit an application where they basically say their findings to date and how they'll test the vaccine out. After this stage, it usually moves on to testing phase. So in phase 1, researchers gather a small amount of human test subjects in order to administer the vaccine onto them to see if it's safe. And then in phase 2, they administer the vaccine onto hundreds of human test subjects to gain more information on safety, immunogenicity, immunization schedule, and dosage. Finally, in phase 3, they administered the vaccine onto thousands of human subjects in order to ensure maximum safety and effectiveness. After all these testing phases, the vaccine still has to go through approval, manufacturing, and finally quality control in order to ensure that the vaccine is actually performing the way it was expected to perform. The fourth topic that I'll be discussing is vaccine development for COVID-19 specifically. So on May 3rd, the EU member states, Japan, Canada, Saudi Arabia, Norway, and the UK basically spent $8 billion for the development of a successful COVID-19 vaccine and to ensure effective global access. At the same time, the US basically created this project known as Operation Warp Speed, where they basically spent several $billion to develop enough doses for the US population. On top of this, there are several private companies that are going ahead and creating more vaccines. An example is BioNTech, who basically partnered with Pfizer to develop mRNA vaccines, and their phase 1 or phase 2 trials started at the end of April, and US patients were dosed in May. Oxford University's Jenner Institution partnered with AstraZeneca to develop and produce a viral vector vaccine. Their phase 1 or 2 trials started at the end of March, and if positive animal results replicate in humans, then phase 2 or 3 can start in June. Lastly, Moderna is currently running animal studies with humans. These rapid developments are increasing the chance of a successful vaccine and leading to immunizing the population. However, the timeframe still remains unclear because the successful vaccines will be first given to healthcare workers and the high-risk population. On top of that, a bigger challenge is that the techniques used for COVID-19 vaccines, which are predominantly DNA and RNA vaccines, they have not yet been proven to work in humans. The final topic that I'll be discussing in this episode is drug repurposing. So drug repurposing is a term that is used to define the process of finding a new purpose for a drug that was basically used for another disease. So in this context, an existing drug that was used to cure another disease is used in studies to see if it would cure COVID-19. Drug repurposing is useful because it's a much more efficient process than creating a drug from scratch for a new disease like COVID-19. An example is remdesivir, which was originally used for Ebola, and chloroquine, which was originally used for malaria. So that concludes my third episode of this series. As always, if you guys have any questions, comments, or thoughts, please feel free to leave them down below. We are finally done with the scientific aspect of the pandemic, so we'll be moving on to other aspects of the coronavirus pandemic in the later episodes. Until next time, see you. Bye.